Good day, everyone. Our lesson for today is all about somite formation. But before we proceed to our discussion proper, we must first know the objectives. So at the end of this lesson, we will be able to understand the definition of somites, their different types and the components of mesoderm, and also to analyze the formation of somites and how it works. So what is somite? Somite is a division of the body of an animal or embryo. They are precursor populations of cells that give rise to important structures associated with a vertebrate body plan. When we say body plan or an animal body plan, it is the sum of the organization and combination of an animal's body parts and features, including how a body is segmented, its body symmetry, and its disposition of limbs. Also, somites are bilaterally paired blocks of paraxial mesoderm that form along the head-to-tail axis of the developing embryo in segmented animals. Overview of somite. Somites are transient structures found in the embryo. They develop from the mesoderm, located adjacent to the notochord, or also known as the paraxial mesoderm, and are formed in pairs. The somite pair formation begins at the caudal. When we say caudal, it is moving away from the head. It begins at the caudal or head end and continues towards the tail. Each somite comprises of an outer dermomyotome that is derived from the epithelial cell layer and an inner sclerotome that is formed from a mesenchymal core. The dermomyotome divides into a dorsal dermatome and a ventral myotome. The sclerotome forms the intervertebral discs, the dermatome forms the dermis of the skin, and the myotome forms the muscles of the limb, abdomen, pelvic girdle, and chest. The somite number is unique for each species and can be used to determine the developmental stage of the embryo. So here's an example of species and their somite number. For human, there are 44 somite number. For mouse, there are 65. For chicken, there are 55. For lizard, there are 72 to 73. For xenopus, there are 42. And for zebrafish, there are 32 somite number. So now I will discuss to you how somite forms. This is the embryo or the ectodermal layer. If we peel this off, we can see a simple undifferentiated flat layer. So here is the mesoderm. Along with this neural tube is the notochord. The notochord is also called as the chordomesoderm or the axial mesoderm because as you can see, it is lying in the central axis. This part here, which is running just parallel to the notochord, is called as the paraxial mesoderm. This part here is the lateral plate mesoderm. And lastly, this part here is the intermediate mesoderm. So those are the components of mesoderm. Mesoderm. Mesoderm is a germ layer that develops during gastrulation in the early development of the embryo of most animals. To understand more about the components of mesoderm, which I have discussed earlier, here are their definitions. First is the lateral plate mesoderm. The lateral plate mesoderm gives rise to the appendicular skeleton. The appendicular skeleton includes shoulder girdle, pelvis, upper and lower extremities. It forms the progenitor cells. Progenitor cells are considered to be the adult stage of stem cells, but they reside in a stage of further differentiation and they divide only a limited number of times. They constitute the heart and cardiovascular system, blood, kidneys, smooth muscle lineage, and limb skeleton in the developing vertebrate embryo. Another component of mesoderm is the paraxial mesoderm, which gives rise to the axial skeleton. The axial skeleton includes bones in skull like cranial and facial bones. And lastly is the intermediate mesoderm, which generates the urogenital system like the kidneys, gonads, and their respective duct system. So we all know that spinal cord is the most important and delicate structure of the body. That is why it is encased by the vertebral column in order to protect this structure. This vertebral column is made up of small segments called vertebrae, and they are attached together to form the vertebral column. As what I have mentioned in the previous slide, the vertebral column is segmented, and so paraxial mesoderm becomes also segmented, and as they become segmented, they will form the vertebral column along with the associated muscle as the skin of total area. These segments of paraxial mesoderm are called somites. So these are the somites, and this is how it forms. Here. This formation of somites from the paraxial mesoderm are called somitogenesis. So somitogenesis is the process by which somite forms. And now we can see the cross-sectional view of mesoderm. So now we can see an irregularly shaped mesenchymal cells interspersed between the ground substance. They become close together and they acquire a sort of world arrangement of these mesenchymal cells. And then the outer mesenchymal cells undergo a mesenchymal to epithelial transition. So now I will discuss to you the types of somites. For instance, these are a group of people. And then one of those people will say that, okay, you 5 people, you have to form the dermatome. And then you 3 people, you have to form the myotome. And the remaining people, you will form the sclerotome. Those that I have mentioned are the types of somites. The difference between epithelial cells and mesenchymal cells is that epithelial cells are differentiated to cover body surfaces, line body cavities, and hollow organs, while mesenchymal cells are differentiated into a variety of mature cell types such as connective tissue, cartilage, adipose tissue, and lymphatic tissue. These are the types of somites, and I have discussed this earlier, which I gave an example. And the first type of somite is the dermatome. The dermatome is the dorsal portion of the paraxial mesoderm, which gives rise to the dermis of the dorsal skin. Another type of somite is the myotome. The myotome is the part of the somite that forms the muscle of the animal. The epaxial and epaxial is under myotome, and the difference between the two is that epaxial is the part where it develops and forms the extensors of the dorsal part of the vertebral column, while epaxial gives rise to the intercostal muscles, which are muscles that are present within the rib cage. The last type of somite is the sclerotome. It gives rise to the vertebrae and associated ribs, tendons, and other tissues such as the vascular cells of the dorsal aorta and intervertebral blood vessels. So those 3 are the types of somites. So to further understand about the somite formation and their types, I will show you a video about how somite formation works.
Somite formation. The picture on the right-hand side shows a 31-day-old embryo in the lateral view, and in green we can see the head and the 6 branchial arches, and then along the dorsum we see in yellow these segments of somites. And there's the cervical somites and thoracic and lumbar somites, as well as sacral somites. And then we have at the tail end some of these coccygeal and then more cranially pre- and post-otic somites that we're not going to really consider. All together in yellow and purple, there's 40 to 42 somites in humans. We're going to focus on the somites highlighted in those dotted lines, so the 8 cervical, 12 thoracic, 5 lumbar, and 5 sacral somites. And we're going to take one of those segments, take a cross-section through it, and ta-da! There we have this cross-section through a somite, and one somite highlighted in yellow. And in this picture, there's the neural tube will become the spinal cord. Neural crest cells become the dorsal root ganglion. And simply for orientation, there's the gut tube with the associated coelomic cavity, and all on the other side, Shing, there we've got the somite. So let's talk about the dermatome, myotome, and sclerotomal components, those three elements that comprise these condensations of periaxial mesoderm that we know as the somites. So first in pink, we have the dermatome. These dermatomal cells are more superficial in this somite, and they're going to migrate out and become the dermis of the skin of the back. The overlying ectoderm is derived, it becomes the epithelium. The dermatome becomes the skin of the back. And as you go forward more and you get this ring, as you can see in this picture in pink, the body wall and the skin of the limbs comes really from the somatic part of the lateral plate mesoderm, but it's from the same segment, so the principle's the same. And there we have in pink this ring of skin. And in yellow, there's our spinal nerves. And so the dorsal root, the dorsal ramus primarily, sends off branches to provide sensory innervation of the dermis of the dorsum of the back. And then this ventral ramus courses and gives rise to a lateral branch that provides sensory innervation of the lateral part of this band of dermis, and then it terminates as an anterior cutaneous branch, which then sends sensory information from the front. And therefore, when we see this entire band in pink, it's innervated by all the branches bilaterally of dorsal and ventral rami, and it forms this ring of innervation of the skin. Hence the clinical definition of a dermatome, an area of skin supplied by a single spinal nerve level. This segmental happens 8 times in cervical, 12 thoracic, and so forth. Myotome is the region highlighted in purple and red because the myotome has 2 parts. The epimere, which are these cells that migrate more dorsally to the epiphora pons, the top part of this developing embryo, and these cells are going to develop into the erector spinae muscles. Whereas the hypomere in red, these cells migrate more ventrolaterally and they form the body wall muscles, hypo below on the bottom of this developing embryo, but in the cervical and lumbosacral regions becomes muscles of the limbs. The yellow arrow shows these, epimere becomes these epaxial muscles. And the dorsal ramus, that dorsal branch, will innervate not only the skin of the back, but also these epaxial muscles like erector spinae. In the hypomere, these cells form the hypaxial muscles, which is 4 layers of lateral body wall, a subvertebral group of muscles, and the ventral strap muscles. And as I mentioned before, in the cervical and lumbosacral region, myotome, hypomere components of the myotomes become muscles of the limbs. The ventral ramus branch will innervate all muscles of the body wall and the upper and the lower limb muscles. Finally, in green, there we have the sclerotome, and these sclerotomal cells will migrate and surround this developing neural tube, which will become the spinal cord. And so there we've got that neural tube surrounded by these bony segmental elements we know as vertebrae with these costal components. And these are segmentally from top to bottom, and so they protect the spinal cord, but there's spaces in between for the spinal nerves to exit at each segmental level, and also allows for some flexibility. So here we have that picture now, highlighted with the dermatome, epaxial, hypaxial muscles, and the sclerotomal component. Looking at this lateral view yet again, and we take one segment and shing, we blow that up, and there we have a segment. All those components of vertebral, spinal cord, spinal nerve levels with dermatomes and myotomes. But how about this level? Same thing. At that level, there is a vertebral level, spinal cord level, associated spinal nerve with dermatome and myotome. Oh, what about this level? And the same thing. The key to this is to recognize that every single segmental level of 8 cervical, 12 thoracic, 5 lumbar, 5 sacral levels, there is going to be a vertebral element. There's a vertebral element. There's going to be a spinal cord element. There's going to be a spinal nerve element with a dermatome and myotome. Those 3 themes we're going to see over and over again now as we look at the neurological levels of each of these regions.
So that's how somite formation works. Somites are precursor populations of cells that give rise to important structures associated with the vertebrate body plan and will eventually differentiate into dermis, skeletal muscle, cartilage, tendons, and vertebrae. The somite formation begins as paraxial mesoderm cells become organized into whorls of cells called somitomeres. The somitomeres become compacted and bound together by epithelium, and they will eventually separate from their presomitic paraxial mesoderm to form individual somites. So that ends my presentation, and I hope you learned something about my discussion. Thank you.